TARC is a unique structure insofar as the state of Texas has really emphasized that learning more about Alzheimer's disease, diagnosing it, and treating it for the citizens of Texas is an important goal, to bring together the multiple institutions in Texas who are doing work on Alzheimer's disease.
TARC involves all of the top 10 medical schools in the state, and that encourages collaboration across institutions as well. So one of the ideas of TARC was to help us get out of our academic silos, if you will, and find out what other investigators in our own state are doing.
Research is a team science or team sport. No one individual can do it all. We partner up with other individuals with different skill sets and specialties and look at things from different angles.
Having everyone at the table at the same time allows you to see things from angles that you might not otherwise have seen. And it's helpful to think not only about the moment that you're working in or the specific thing you're focused on, but how it fits into the bigger picture.
The kinds of problems that we need to solve are very, very complex. They're beyond the scope quite often of one laboratory or one investigator. And sharing resources, sharing ideas and methods is really the way to come up with the most effective answers to some of these really challenging problems.
This kind of work is not like a terminal project that has a defined start and a defined stop. Because one discovery rolls out and that discovery opens the door to something very useful.
It's always amazing to me how people can see the same set of data and find different things interesting or worthy of study, and that's great because nobody has all the answers.
The collaboration aspect allows us to do more than we can do at our site alone. Together we can accomplish much more in more diverse populations with a bigger impact.
It takes a village, it takes a state, it takes a country to fight something that is so challenging and has so many ramifications.
It's a formidable force in research if you're interested in the dementias. So TARC is just going to continue to grow, not only in the research that it produces, but also in its understanding of what needs to be at the forefront of research and what needs to be the priorities of researchers in this field of Alzheimer's disease and related dementias.
And together, we're stronger and better than any one of us alone.
Welcome to the 2025 TARC Scientific Symposium. Please welcome the Scientific Director of the Texas Alzheimer's Research and Care Consortium, Dr. Monroe Cullum, from UT Southwestern Medical School.
Good morning and welcome to TARC 2025. It's delightful to be here, and it's a very exciting time to be here as well. I've got some comments and some data bits of my own about TARC that I think will be informative for the group. But I really want to thank all of our institutions. I want to thank our External Advisory Committee for keeping us rolling. And without further ado, I'll just remind everybody of our participating TARC sites, if you will. We were pleased to add UT Tyler this last year. That was approved by the Texas Alzheimer's Council. So we now have all the medical schools in the state pretty much involved, and each has a representative. So if depending on which institution you're at, know your representative. Those steering committee members, part of their job is to encourage grant applications for our grant cycles. We've had several rotations in and out. We welcomed Giulio Tagliatella, Isam Saloom, and Andrew Schmidt from their respective institutions who are now members of our steering committee. So I put that up to remind you and let you know you have steering committee members. So if you have questions about TARC, they're a good source of information. Well, TARC continues to grow in visibility and impact, and I'll show some statistics about that in a moment. But we really are helping to advance not just research from the basic to the clinical sciences, but also helping to advance careers and develop young talent across our institutions, as well as encouraging collaboration. And of course, the state legislature is meeting now. TARC is funded every 2 years. And knock on wood, we hope they see us favorably again. But we've had a wonderful return on investment. This last year with the RFA was to target junior investigator awards to encourage that. As you all may know, the process is we have external reviewers outside the state that review our grants. Those reviews are tallied and summarized, and then they go to our external advisory committee for recommendations. But I also want to remind people we have a TARC data set that's available for researchers as well. So all you have to do is send us an email if you're wanting access to our database. TARC started out as a multi-site collaborative clinical research endeavor, with each site collecting research subjects and biospecimens. Those data all still exist. We have actually, I think it's closer to roughly 3,000 cases. So we have a very rich database that can be used for mining for papers, projects, posters, as well as for TARC grants, which we always like to see. If you go to the TARC website, you can find that information about how to do a data request. It's not totally open per se. We do make you make a brief proposal and rationale, but it's a pretty painless process. So this last year, we received 73 grant applications from most of the TARC institutions, and we had to select from those. Our reviewers uniformly said this was the most impressive group of applications that they had seen to date. We do rotate our external reviewers, but this was a pretty consistent comment. So they felt like there were many worthy applications that could not be funded due to our budget limitations. But the expert group that we have as our external advisory panel, led by Dr. Ron Peterson, you saw Doug Galasco, Jerry Schellenberg, Todd Goldie, and Christine Yaffe, all experts in their areas. They've been advising us over the years. Sometimes they advise us to make some tough decisions, and sometimes they pat us on the back and have recommendations as to just how to continue to evolve and improve. So we made $7 million in awards this past year to 21 different projects. As I say, we had at least 10 more that were fantastic sounding projects that we hope went on to be funded by other sources, or that you're retooling and are going to resubmit. Actually, we love to hear from people when, for example, your TARC, especially when your TARC grant is funded as pilot, then you get your NIH grant. We love to hear about that, and we do try to track that. But even if your TARC grant was not funded, we love to hear about the projects that, hey, I didn't get the pilot there, but I got this R01 from NIH or whatever it was. And we do hear that, so we know we're turning down really good applications. But we focused on the junior investigators. We had a couple of collaborative grants. We do try to give a little priority to use of TARC data, but it's not necessary. You can use other data sets, in fact, and mine archival data if your science questions are good. And then we gave out 5 more senior established investigator awards as well. But we do like to see collaborative grants that involve more than one TARC site. So if you go back in TARC's history, so I mentioned earlier, TARC was originally a multi-site data collection operation, much like the National Alzheimer's Coordinating Center. And then in 2018, we switched to a grant funding mechanism based on input from the EAC and other leaders around the state, including the Texas Alzheimer's Council. And since then, we've given out roughly $30 million in grants to Texas researchers. If we look at the number of grants and the amounts generated by those investigators who had originally been funded by TARC, the number is about 5.5 times that. So this is the information that we are so proud to see, and this is what we want to see. This is what TARC is all about, is stimulating, encouraging, helping build careers in research in Texas. It's a great time and an exciting time to be a researcher in dementia, and it's an exciting time to be a dementia researcher in Texas as well. The bottom point there is on the dementia prevention initiative for Institute for Texas, the DPRIT it's called. Everybody's probably heard of CPRIT, the Cancer Prevention Initiative that was funded by our taxes, actually, some years ago. The DPRIT is a dementia-focused bill, House bill related to House Bill 132, that's currently, I think it was looked at yesterday. It's got good momentum from what I hear from my sources. We're hopeful. It will be a $3 billion initiative that should be, if approved, will go out to voters in the fall. And if it's approved, it would be enacted next year. So we like to think that TARC has played a small role in that, if that comes to fruition, in terms of demonstrating not only the collaboration across the state, but the research excellence that is Texas. As part of those developments, obviously UT San Antonio Health Science Center got their NIH-funded Alzheimer's Disease Research Center, I think about 3 or 4 years ago, which was fantastic, and at the time the only funded ADRC in the state. There's now a North Texas Alzheimer's Disease Research Center application that's being looked at hopefully by NIH as we speak, actually. And I mentioned DPRIT, which has massive implications for all of our institutions. So I would definitely encourage everyone to be thinking about projects, endeavors, research grants, postdoctoral training grants that might be good for TARC funding in this next cycle. Our symposium has continued to grow impressively over the years, too. We've got a number of abstracts accepted in blue, and as you see in orange there, we had over 400 people register to attend. Now, weather conditions, that lovely, bizarre Houston and Galveston weather has kept a number of people away, unfortunately. We've got a few intrepid souls here, though, so thank you all for making your way, putting on your snow boots, your Texas snow boots, and making your way up the freeway. But the Texas Council and the legislature love to see these numbers, so we really do mean it when we say thank you for coming, and we hope you have a great meeting as well. We've got a busy day ahead. And I've got a lot of people to thank. I'm going to probably forget some. I already thanked you for attending, and our steering committee, our advisory committee. But our TARC staff has just been absolutely phenomenal. There was a retirement. You're not going to see Bruce Jones running around our meeting this year. Bruce retired, despite all my begging. But we have a wonderful replacement, Elita Godby. You just want to stand so people can see who you are, Elita? Ms. Godby has replaced Bruce Jones, and of course, Nikita G. Is Nikita in the? She's probably out running around and busy, but got to say hi to Nikita if you see her. She's really been our conference chair, our conference organizer. But I also want to thank the entire team of students and volunteers and research staff that we brought with us from Dallas, as well as Amy Scott, John DeSilva, and Ben Porter, who's up here with this is not one of those neurosurgical contraptions. He's okay, it's for his camera, so, or so he tells me. But it's just an extremely exciting time for TARC. I do want to encourage any and all of you, if you have a chance to talk with your governmental affairs people, the leaders at your institutions, make sure you're mentioning TARC and talking about TARC. That really helps. When they're hearing about us from different angles and different institutions, different sources, that provides the momentum and the buzz, if you will, about TARC. And we actually were very, very fortunate. We had the governor's wife join us for a little while yesterday. So she's aware of TARC, and it's something that we're looking forward to, a really terrific meeting as well. And I think those are all my opening words. I would like to turn it over to the vice chair of the steering committee for TARC, Dr. Rodrigo Morales.
All right, so welcome to this session. I have to say that this is has been one of the attendees' favorite sessions. You know, we will hear about updates on the different sites. And I want to, before we start, I want to second on something that Munro just mentioned about the TARC grant application. So as soon as the notice is released, please ask about it. You have any question, go to your institution representative on the steering committee and try to apply. We encourage you to apply. I think that is a good opportunity, and I'm talking personally from my institution. I know that TARC funding have helped to secure additional funding and boost the career of young investigators. So please ask for that and please apply. So now we will start with this data please session. And the first speaker we have is Ms. Shana Garza from UTRGV.
Hi, my name is Shana Garza. I'm a master's student at the University of Texas, Rio Grande Valley, and today I'll be presenting research on blood-based and MRI markers of Alzheimer's disease in Mexican Americans. Sorry about that. So currently, Alzheimer's disease is diagnosed through the use of various tests, including PET scans and collection of cerebrospinal fluid. However, these tests are often expensive and inaccessible to many, including Hispanics, which are about 1.5 times more likely to develop Alzheimer's disease compared to non-Hispanic whites. One way to hopefully increase the use, to increase minority participation in Alzheimer's research is through blood biomarkers, which have proven to be cost-effective, minimally invasive, highly accurate, and more accessible compared to alternatives. However, few have investigated these blood biomarkers in relation to MRI 80 signature regions, especially in Hispanics, leaving a gap in research. So we help to fill this gap by studying the association of baseline Alzheimer's disease and related dementia, or ADRD, blood biomarkers with MRI 80 signature regions using a Mexican-American population-based cohort. Our objectives were to better understand these ADRD blood biomarkers in Mexican Americans and to examine the genetic effects and non-genetic effects, which would be the blood biomarkers in this case, on 80 signature regions. So for this, we used 1,044 participants between the ages of 18 to 85 years old from the genetics, brain structure and function cohort, also known as GOBS. And this cohort is part of a larger study known as the San Antonio Mexican American Family Study, in which all the participants were Mexican Americans coming from the community of the San Antonio area and came from large families. And from this GOBS cohort, we included participants that had data on brain MRI imaging, genotyping of the ApoE4 allele, and ADRD blood biomarker data. So in our methods, we used a linear mixed regression model, which we ran in statistical software known as SOLAR. And for our covariates, we used age, sex, the ApoE4 genotype, and the estimated intracranial volume. And since the participants in the study are related to one another, we accounted for this using the linear mixed effects model, which is built within SOLAR software. But before we ran our analysis, we first had to normalize the raw data of the blood biomarkers, which can be seen on the top since the data was heavily skewed to the right in the top graph. And then after normalizing this data, we get the graph shown below showing a normal distribution. So in this cohort of the 1,044 participants, the average age was 41 years old. 131 participants had at least one APOE4 allele. 605 participants were female, 14 had dementia, 17 participants above the age of 60 or 60 years old or older had mild cognitive impairment, and 42 participants below the age of 60 had MCI. And there were a total of 73 families in this cohort. So for our variables, we used the exposure variables we used were the blood biomarkers, including NFL, GFAP, total tau, beta amyloid 40, 42, and 42/40. And our outcome variables consisted of cortical and subcortical brain regions associated with Alzheimer's disease. So for the 7 cortical regions we used were based on Dickerson and colleagues' previous research, which showed that brain regions certain brain regions became smaller 10 years before Alzheimer's symptom onset, and those regions are highlighted above. We also included an additional 80 signature region, the fusiform from previous research of Jack and colleagues, and we added up the 780 signature regions from the Dickerson and colleagues research to create an 80 signature value. So from our findings, we found that NFL was associated with 5 out of 7 of the cortical 80 signature regions, even when accounting for heritability as shown in Table 2. And NFL was also associated with the overall 80 signature value and with 2 of the subcortical brain regions, which were the hippocampus and the amygdala. And the pie chart helps show that the proportion of phenotypic variance, which in this case would be the cortical thickness of the 80 signature value, the heritability helps show that about 50% of the phenotypic variance is due to genetic factors, while 1.44% was due to all the covariates, including NFL. And out of this 1.44%, 1% of the phenotypic variance for this trait was due to NFL. And so our findings for the other ADRD blood biomarkers, we found that there were significant associations with brain regions involved in facial recognition, including the fusiform gyrus and the inferior temporal lobe. Specifically, GFAP was associated with the inferior temporal gyrus, and GFAP and beta amyloid 42 were both associated with the fusiform. We are further analyzing these results to see if there's, with genetic information, to see if there's a SNP that is commonly expressed in both these blood biomarkers and the brain regions. So based on our results, we concluded that blood biomarkers of Alzheimer's disease and related dementias were associated to MRI 80 signature regions in this community-based cohort of Mexican-American families. And we further identified NFL being the main blood biomarker associated with these 80 signature regions. And this research is important because it helps, enhance clinical utility by improving blood biomarker cutoff levels. It gives us a better understanding of the association between brain imaging and blood biomarkers, specifically in Mexican Americans, has implications for better brain health by giving us a better understanding of healthy brain aging in Mexican Americans, and can hopefully help improve diagnostic criteria of Alzheimer's disease and related dementias in Mexican Americans. All right, thank you so much for the invitation, and hope you enjoyed the rest of the conference.
Something I forgot to mention, we will have no questions during the session. You are welcome to ask the speakers during all the breaks that we will have during the symposium. So our next speaker is Dr. Xia Xuan Fu from Baylor College of Medicine.
Hi, oops. Oh, let me go back. Good morning, everyone. My name is Annie Fu, and I'm a postdoc at Baylor College of Medicine with Dr. Jeannie Chen. I'm excited to be here this morning to share with you all our work on identifying factors that might confer resilience or susceptibility to cognitive impairment in Alzheimer's disease. It's been found that nearly 1/3 of individuals with amyloid pathology in their brains remain non-demented throughout their lifespan, suggesting cognitive resilience to disease. Notably, in a mouse model of amyloid pathology, we also observed a similar differential resilience and susceptibility to cognitive deficits, such that some mice also appear to be resilient to disease. So in these APP mice, by 1 to 2 months of age, they all start off by exhibiting spontaneous seizure activity that is similar to what is observed in AD patients. Most mice then continue along this path. Oops. Oh my gosh. Most mice then continue along this path and develop severe memory deficits and eventually plaque deposition. However, we noticed that about 1/4 of these mice seem to phenotypically diverge around 3 to 4 months of age, such that they stop having seizures and are cognitively normal, despite going on to develop a similar level of plaque deposition as their susceptible littermates. And so we wanted to take advantage of this natural phenotypic difference that these mice exhibit to identify resilience or susceptibility factors. And so to sort mice into resilient and susceptible groups, we, with as little prior manipulation as possible, we turned to delta phosphate as a proxy. So delta phosphate is an activity-dependent transcription factor that is also a marker of a history of seizure activity. You can see here that non-transgenic mice express low baseline levels of delta phosphate in their hippocampus. APP mice that become resilient also express low non-transgenic levels of delta phosphate. However, APP mice that are susceptible continue to have seizures and thus have high levels of delta phosphate. Delta phosphate expression tightly corresponds to the number of seizures mice are having per day, and decreasing seizure activity with an anti-seizure drug reduces delta phosphate expression. Both resilient and susceptible APP mice express similar levels of APP, suggesting that this phenotypic difference likely isn't due to copy number variation of the transgene. Importantly, increased delta phosphate expression is associated with worse performance in the Morris-Water maze, which is a hippocampus-dependent memory task. And we found a similar relationship in human patients. So in individuals with mild cognitive impairment, which is thought to be prodromal AD, increased delta phosphate was also corresponded with worse mini mental state examination scores. And so together, these results tell us that delta phosphate can be used as a proxy marker for resilience or susceptibility to seizures and cognitive impairments in disease. And so, to study resilience and susceptibility, we decided to focus on this 3 to 4 month time point, which is right around when APP mice diverge into resilient and susceptible groups. We grouped the mice based on their delta phosphate expression and performed bulk RNA sequencing on their hippocampal dentate gyrus, which is a brain region that is notably highly resilient to cell death in both AD and epilepsy. And so it seemed like a good place to look for resilience factors. Using differential gene expression analysis, we identified lists of genes that were differentially expressed between susceptible and resilient APP mice, shown in pink, between non-transgenic and susceptible APP mice, shown in yellow, and between non-transgenic and resilient APP mice, shown in blue. And if we look at the overlap between these gene lists, we see that there is a group of 73 genes that are distinctly differentially expressed in the resilient mice compared with either non-transgenic or the susceptible APP mice, meaning that their expression level is unique in the resilient mice compared with the other two groups. And so we hypothesized that these may represent resilience factors. There's also a large group of 797 genes that are distinctly differentially expressed in the susceptible APP mice compared with the other two groups, which might represent susceptibility factors. But how might these genes be conferring resilience or susceptibility? So to get an idea at the functions of these genes, we performed a gene ontology analysis using Klugo. And so we can see here that the resilience genes are involved in functions such as ion transport, neurogenesis, cell adhesion and signaling, as well as some glial functions. The susceptibility genes are also involved in a lot of the same general categories. However, the individual clusters within each general category differs between the resilience and the susceptibility groups of genes. And the susceptibility genes are also involved in some metabolism and synthesis functions. So we're currently using these plus other analyses to delve deeper into understanding what makes resilient and susceptible APP mice different from each other, as well as to help us narrow down and prioritize genes of interest to investigate in further hypothesis-based testing. And so, for example, we would predict that if we overexpress resilience factors in APP mice, we'd be able to bias more of them to become resilient. Whereas if we overexpress susceptibility factors, we'd bias more of them to become susceptible. And so, in summary, we took advantage of this natural resilience that some APP mice develop to identify candidate factors that we can manipulate to maybe tip the scale towards resilience and away from susceptibility. Current study in the lab looking at genes that were identified through this method indicate that we are indeed able to enhance resilience and reduce susceptibility in our APP mice. And of course, the ultimate goal is to eventually translate these findings into treatments to promote resilience and reduce susceptibility to cognitive impairments in human individuals with Alzheimer's disease. And with that, I'd like to end by thanking all the members of our wonderful lab, as well as our funding sources, and also thank you to all of you for your attention today.
Good morning, everyone. My name is Alyssa Aguirre, and I'm so honored to be here today. It's wonderful to see all of you as a part of the TARC family. I was hired at the University of Texas in Austin 8 years ago to be the research coordinator for our first TARC project. I now serve as the Assistant Director of Dementia Care Transformation, and I'm excited to share with you the findings of our latest research today. Other speakers help me out, there we go. All right. I'd like to first, of course, acknowledge our wonderful research team, especially Dr. Jared Bench, who is here today, who is a phenomenal research mentor, as well as Rovi Ann Tindan, our research coordinator, and Andrew Sampson, our undergraduate research assistant. I'd like to start out by thinking about how do we determine our research questions? Often they're shaped by the priorities of funding agencies, sometimes our organizational missions, or collaborations with colleagues. However, sometimes the most meaningful research questions emerge organically out of gaps identified in clinical practice. 8 years ago, we designed the Comprehensive Memory Center with both diagnostic and care pathways. The care pathway is focused on providing one-on-one counseling to caregivers for support. As we reviewed outcome measures for caregivers, we noticed an interesting pattern. The majority of them focused on depression, anxiety, strain, and burden. And while measuring caregiver mental health is important, it doesn't necessarily capture the needs and priorities that caregivers themselves want to prioritize in clinical settings. Myself and my social work colleagues set out to find a tool that really assessed the needs and goals of caregivers that could be implemented in clinical practice. Surprisingly, we really weren't able to find such a measure in the literature. This is not a minor problem. According to the Alzheimer's Association, 11 million Americans provide unpaid care to persons living with dementia every year. We know that caregivers need more assistance with care coordination and navigating the healthcare system. We weren't alone in recognizing this gap. The research community has called for the development of outcome measures that go beyond the deficit-focused framework, finding that the majority assess depression and burden, and those are the primary outcomes for evidence-based caregiver interventions. However, limited progress has been made in this area to develop new tools. Lauren Bangerter and her team provided recommendations for the future development of outcome measures. Those recommendations including incorporating feedback from both clinicians and caregivers, using a mixed methods approach, and making sure they're brief and self-reported for clinical use. Our team addressed this gap by creating the Coping, an assessment tool designed to measure the diverse priorities of caregivers in clinical settings. Our process began with an initial refinement stage of 16 items. We recruited dementia professionals, around 24 from around the country, as well as 28 dementia caregivers. We assessed reliability by having participants take the Coping first at baseline and again 2 weeks later. Our team was also interested if the traditional metrics of psychosocial burden and anxiety could predict caregiver needs and goals. All 16 items were rated as important by both caregivers and dementia professionals. They gave us wonderful feedback on the measure, including simplifying the language, such as using the term dementia instead of cognitive impairment, combining 2 items that were similar, as well as providing concrete examples for each item. The newly revised 15-item Coping was used in the subsequent phase. For this phase, we recruited an additional 100 caregivers, and they completed the Coping at baseline and 14-day follow-up. They also took psychosocial measures such as the ZERIT, the positive aspects of caregiving, geriatric depression scale, and the quick dementia rating system. In this sample of 100 caregivers, they represented a diverse group. They were representing 8 different types of dementia as well as mild cognitive impairment. 40% identified as non-spousal, 18% identified as Hispanic or non-white, and 25% identified as male. These are 6 sample items from the Coping questionnaire. We asked the caregivers to first rate which items were important to them and indicate which ones were a top priority. Let's take a look at some of the results. For the importance ratings, they range from 2.2 with basic needs to over 4 with learning more about dementia, psychosocial support, and learning more communication strategies being rated the most important by caregivers in our sample. When we think about top priorities, we know that caregivers are influenced by a variety of factors, such as their familial relationship with the care recipient, their prior life experiences, the timeliness of the diagnosis, as well as availability of other sources of support. What we found in our sample is that the needs and priorities were quite varied throughout our sample, but no single item was rated as a top priority by fewer than 5% of our sample. The top 3 priorities identified by caregivers were psychosocial support, finding activities for the person living with dementia, and increasing their dementia knowledge. Exploratory factor analysis revealed 4 kind of main themes or clusters of items on the Coping. The first theme was pragmatic needs, caregivers who were interested in learning more about finding help in the home, assistance with long-term care housing, help with legal paperwork, transportation options. The second factor grouped items around knowledge and understanding, caregivers who were interested in learning more about dementia, strategies to improve their own brain health, as well as psychosocial support for themselves. The third factor was around behavioral management, caregivers who really wanted assistance with managing difficult behaviors, as well as learning new communication strategies with their loved one. And finally, the fourth theme was about interest in research and, to a lesser extent, caregiver programs. Thinking about how these themes might present themselves in clinical practice, here are some of the things you might hear from caregivers. I'd like to take us back to our research question. How do we measure the needs and goals of caregivers? As you recall, our team was interested in finding if traditional metrics of anxiety and burden can predict caregiver needs and goals. What we found is that demographic factors, dementia type, severity, anxiety, and burden only accounted for 10 to 15% of the variance in our sample. So what does that mean? Our findings really challenge the notion that we can make assumptions about caregivers based on their clinical profile. A caregiver caring for someone with behavioral variant FTD may not rate managing behaviors as their top priority. In our study, our top priorities were psychosocial support, activities for the person living with dementia, and increasing their knowledge of dementia. Of course, tools that are brief and self-reported are going to have higher utility in clinical settings, and we were pleased that in our study sample, caregivers were able to complete the coping in under 3 minutes. Some of our study limitations include our sample size, as well as particularly regarding historically underrepresented groups. We're really excited about the future of the coping. Some of the things that are happening right now is that a group of community health workers have translated the coping into Spanish and are using it in the community here in Austin, as well as a group in North Carolina using the coping as a part of their guide model to assess caregiver needs, and a group in the Midwest using the coping as a part of their extension of the Care Ecosystem Project. We're also hoping to expand the coping to other neurodegenerative conditions, such as ALS and Parkinson's, and make sure that it remains relevant. For example, potentially adding an item such as biomarker education for caregivers. Thank you so much for all of your attention today, and I am excited to learn along with you with the rest of the presenters.
Now we will hear from Dr. from UT Health Houston.
Hi. So today I'm going to talk about, my name is Asaf Gottlieb. I'm going to talk a pilot study that we conducted. It was recently published using fitness trackers to predict mild cognitive impairment status. It was done with other people from my school, Chidi Xu and Yejin Kim, and with Dr. Paul Schultz, who recruited the people from the Government School of Medicine, and recruited the patients, the participants in the study in his clinic, and was funded by the Alzheimer Association grant. So the purpose, sure most of you know, early signs of Alzheimer are difficult to detect. A lot of talks about different markers to do that, and we are aware of that. It allows patients to prepare. It allows testing experimental therapies if it's done early enough, and most of these markers are invasives, slightly invasive or more very invasive. So a pilot study wanted to test if wearable technologies could be used to detect Alzheimer's disease. So the setting is we recruited 12 people with mild cognitive impairment and 8 age-matched controls. Each one was tracked over a course of a month with Fitbits, similar to what I have now. I'm kind of participant number 0. I wasn't part of that, but I was testing the system. I wasn't part of that. I'm not part of the 12 or 8. And we captured activities, sleep, and heart rate, which is what Fitbits provide us easily. We moved that to a classifier and determined which ones were MCI and which one were controls. These are the correct characteristics. You can see that the age is about 70, a little bit imbalance between females in the MCIs versus the controls, but we tested whether that imbalance changes the results, and it doesn't, even if we adjust for gender. You can see that most of the people got around a month, but you see different person. For example, person number 2 sadly returned the Fitbit after a week and said she doesn't want to do it anymore, but most of the people got at least 14 days, as you can see, and some of them got more than a month. And as I'll show you, actually it's fine. Most of these dates are fine, and the number of days to follow. So results, we tested a lot of different combination of all the 3 components that Fitbit captures: activity, heart rate, and sleep. And actually it's main sleep in the night and also naps. So we tested both. The Fitbits knows how to determine which one were naps during the day and which one were the main sleep. We got really good results. I mean, perfect. Actually, when you use all the types, probably owing to the sample size, so we need a bigger sample size to know what's the actual AUC. But as you can see already, when you use 2 out of the 3, you get really decent AUCs, 0.8, 0.9, etc. So you might be wondering, what are the main features that determine which one is MCI, which one is age-matched control? A lot of them are heart rate related, different zones, fat burn, cardio, etc. But the two interesting stuff that we want to follow, which were activity, light activity to be specific, the minutes, the distance, etc., was lower in MCI versus controls. And the deep sleep is something that we found already with the first people that we recruited. They have shorter periods of deep sleep during the night, the MCIs, versus healthy controls. And this is something that I don't think you can easily control yourself. I mean, light activity, someone can tell you, oh, let's go out. Usually the spouses, which were usually part of the controls, but deep sleep is harder to maintain, but maybe with medication or something like that, or therapy, could be tested for causality. We also wanted to know what are the minimal days needed for us to capture from a person in order to get good results. And you can see that already at around 10 days capture, we already get around 0.9 AUC. 20 days, we get much better. So these are the kind of numbers that you're aiming. So basically, after 20 days, it's not adding much. So to conclude, our study suggests it might be possible to use inexpensive, convenient digital markers to detect MCI due to AD pathology. Our hope is that the result will pave the way to early detection. Our initial idea was to do it longitudinally, so we wanted to follow them every year. We managed to get 6 out of the 20 back to the clinic after 1 year, but it's too small sample to show here. And we're looking for additional funding to do that, to increase the sample size, increase the length that we track those individuals, to see whether when they actually cognitive decline, what happens when they cognitive decline, can we capture that early enough? And if we can, we constantly suggest also including other non-invasive markers like plasma and blood that the first speaker, for example, talked about. That's it.
Next is Dr. Rachel Mead from UT Austin.
Hi, I'm Rachel Miss. I'm a postdoctoral fellow in clinical neuropsychology here at UT Austin Dell Medical School. And today I'm very excited to speak with you about an ongoing project we have here at UT Austin that is a collaborative effort between researchers in neuropsychology, neurology, electrical and computer engineering, and speech language sciences. So everyday technologies have the exciting potential to provide a unique window into how our minds are working. So using technologies that are now ubiquitous in our daily lives, like smartphones, we can collect an enormous amount of data covering things like mobility, smartphone interactions, social ability, and more. This type of data collection has several advantages over traditional methods of assessment. We can collect data continuously over long periods of time. We can also collect data from out in the wild, in the real world, instead of the artificial setting of a laboratory or clinical setting. And so this is a promising tool for measuring cognition and everyday functioning in Alzheimer's disease and related dementia research. And so our team here at UT has developed an app that allows us to collect this type of data called Texans, logo up there in the corner. And so we hope that this app will allow us to identify digital biomarkers to identify and predict cognitive and functional decline in folks who might have Alzheimer's or other dementias. And so while this is all very cool and very exciting, this type of work does involve collecting potentially sensitive data. So there are very real reasons why individuals might be concerned about the privacy and security of their digital data. And because this is a relatively novel way of assessing cognitive decline, we don't really know very much about how older adults might feel about the safety and privacy of their digital data, how such concerns might be a barrier to research with these technologies, or how researchers can potentially educate older adults on mitigating digital privacy risk. And so that brings us to the subset of the study that I'm going to speak with you today. So baked into the onboarding of our digital data study is our sub-project in which we hope to characterize older adults' digital privacy concerns and determine whether a brief educational intervention would be effective in reducing these concerns. So as part of onboarding, participants answered questions about their concerns about their digital data privacy in general and specific to the study, as well as their perceptions of their knowledge on how to maintain their general data privacy. Next, we delivered a brief educational intervention that covered the data security measures used in our project. So this covered things like data de-identification, how the data is stored, who has access to it, as well as information specific to the Texans app. And this is an example slide from that presentation. So for example, educating participants that the app might be collecting information about the total number of messages they're sending and the total number of individuals they are contacting, but the app is not collecting information about the identity of folks that they are texting or the actual content of messages they are sending. We also walk participants through how they can elect to turn on or off collection of specific data elements within the app. So participants are able to elect to ultimately not share information about message usage with the app. And so following this training, we then administered those same questions again so that we could look at changes in their responses following the training. And so at the time of preparing this abstract back in December, we had 38 older adults enrolled in our study. That's now almost double that, but this presentation is going to be covering those first 38 participants. And this includes individuals who have a cognitive disorder like MCI or Alzheimer's dementia, as well as healthy older adults as well. And so what we saw at initial onboarding is that participants had greater concern about their digital data privacy in general compared to their concern about their data privacy specific to the study. And then following the intervention, we saw a reduction in participants' concern about their digital data privacy, both specific to the study and in general. So we found a moderate effect size in the reduction of their study-specific digital data concerns and a small to moderate effect size for their general digital data privacy concerns. We did not see any change in participants' perceptions of their knowledge on how to maintain their digital data privacy. So to sum up, older adults in our study had more confidence in the privacy of their study-related digital data despite having concerns about their digital data privacy in general. And this underscores the trust and confidence that our participants do have in us as researchers to maintain the security and privacy of their digital data. We also found that our older adults' privacy concerns can be attenuated through awareness and education efforts by research teams conducting studies using passive digital data monitoring. So this is promising data, but this is an area that is going to require a lot of future study. So we ourselves hope to understand how a diagnosis such as Alzheimer's dementia or MCI might affect folks' concerns about their digital data privacy. But as a field, it's going to be important for us to understand how to effectively communicate with participants about risk of sharing digital data, how to mitigate those risks, as well as how to potentially move these types of interventions to the recruitment stage to reduce the impact of digital data privacy concerns as a barrier to research entry. So as a final takeaway, as medical researchers, as researchers who work with human participants, we are used to thinking about the safety and privacy of our participants' medical data, their cognitive data, their biomarker data. But as we move into the future and incorporate more of these digital tools into Alzheimer's disease research, it's going to be equally essential that we think about the safety and privacy of our participants' digital data as well. So thank you very much to our team who has graciously allowed me to become a part of this project. Thank you to our funding sources, including TARC, who funds my fellowship training here at UT, and a lot of appreciation to our research participants. Thank you.
So I'm not sure if you were hearing me when I was introducing the speaker, so better coming to the microphone. So now we will hear from Dr. Sarah Ross from University of North Texas.
Hi, I'm Sarah Ross. I'm a geriatrician at the University of North Texas Health Sciences Center and the principal investigator for the self-management program for brain health, which was funded through philanthropic dollars from the J.E.S. Edwards Foundation in 2019. Our project is focused on the prevention, on the public health focus area with respect to supporting individuals in making brain healthy lifestyle choices and implementing changes to their daily life that will help them address the modifiable risk factors for Alzheimer's disease and related dementias, in particular with an individual focus, things that might be most relevant to them as an individual, whether that's related to their personal medical problems or family history. And we are also exploring different approaches to health coaching and helping those individuals be motivated and make and maintain brain healthy lifestyle changes, and that includes the beta testing of a application. So we have a traditional health coaching group or arm of our study that is a couple visits every month, one-on-one with a health coach in person, and then we're piloting a application-led or application-supported arm of the brain health coaching intervention. And at the bottom here is the research team, myself. I have a certified health and wellness coach, help from gerontologist and organizational psychologist, and we've had multiple Masters of Public Health interns and medical students who have helped. And just a brief acknowledgement to Dr. Mary Casino, who's a neurologist who applied for the original funding from J.E.S. Edwards for this project. This is a very high-level overview, a simplified overview of what we're doing. We have an online registration and consent process, and the study is open to anyone 18 years of age and older without any cognitive impairment diagnosis. They consent to participate and then complete self-assessment surveys. Then they're part of the intervention, which is brain health coaching. And then after they complete the coaching, they repeat the same surveys. And so the surveys are the items listed here, the 7, what we call in our study, the 7 pillars of brain health. And there's versions of this all over the place, but things like nutrition, social engagement, overall health, exercise, sleep, mindfulness, and outlook. And so individuals are able to do a self-assessment in each of these areas, and then we provide them with a 1-page summary of how they performed in each of those areas, and they utilize that information to make a choice on what they want to focus on during their health coaching. Some people choose one area, some choose 2. What I think is most unique about our program is that they're able to choose goals that are in those health behaviors that are maybe a little bit more abstract, not just eat right and exercise, but I want to work on my focus on outlook and mindfulness. We've had folks set goals of completing a daily gratitude journal. We had somebody who had a goal of learning how to juggle, like a new activity that stimulated their brain and also was a physical activity. Some people who have wanted to get the physical activity with walking, but focus on mindfulness and identifying, things in nature that they're grateful for. So it's been a very unique opportunity to allow people to individualize some of these health behaviors that, again, maybe are a little bit softer. And we've had about half and half, as I listed there, out of the 57 participants that we've served to date that have completed all 3 months of the health coaching. About half of them focus as their primary area on something like diet or exercise, and the other half on some of those other areas that are more the cognitive and mental health behaviors related to dementia prevention. And then I also listed just some themes from the feedback that we've solicited from our participants. They've been appreciative of the program and reported that helped them with their self-accountability and awareness and motivation to meet their health goals, and also just encouragement and optimism to have some support in moving forward with their health goals. And as far as take-home messages, this is, again, kind of the prevention focus. So our group isn't looking for the cure. We're letting other people focus on that. But we need people to focus on the prevention, right? Just what can we do at any age or stage, to help reduce the risk, to hopefully prevent or delay the onset of Alzheimer's disease and related dementias. Our initial cohort was all caregivers, actually. We recruited from a caregiver support group at our institution from people who were very interested, right, and motivated in addressing risk factors to help reduce their risk. And this, again, is unique in that it helps that individual focus on modifiable risk factors that are significant and relevant to them and that they are motivated to work on and improve upon. And so our program helps provide them with the structure that they need to take those proactive steps towards brain healthy lifestyle and disease risk reduction. And as far as our hopes for next directions, I mentioned a digital health application that is being piloted and used, and we're hoping to continue to improve that application and release it to more of a community. It's available to our IRB research study participants at this time, and we're hoping to release it to more of a community availability in the near future. And we're also wanting to invite individuals with mild cognitive impairment and mild stage dementias to participate. At this time, it's just individuals without cognitive impairment, but these types of mitigation strategies and healthy lifestyle behaviors are of course beneficial for those with mild cognitive impairment in early stages of the disease as well. So with that, I'll end and just say thank you so much for the opportunity to participate in the Blitz. Thanks.
Okay. The next speaker is Dr. Diana Yantes from Texas A&M.
Thank you. I realize I'm almost standing between you and a break, so I'll be on time here. My background, I'm a human movement scientist and biomechanist, and a lot of my work is in locomotion and balance in pathological patients and in aging. And specifically, my work is focused on advanced analytics of variability data or fluctuations in patterns of behavior. And our group here, it's a large collaborative group. It includes Dr. Bonnie Scott, who's a cognitive neuropsychologist. Oh, my automatic things are going. I want to go back. Well, you saw their name, so I'll keep going. How about that? All right, so You all are very well aware that it's very difficult to diagnose ADRD at early preclinical stages. And so our long-term goal is really to be able to enhance that early identification and early intervention of those that are on a trajectory towards cognitive decline and functional decline. And we want to go beyond baseline measures, beyond just an average capture of their behavior, and look more at capacity and variability. So one of the things we're using is something we like to call reserve. We have a very general definition as maximal threshold for increased resources just to meet that short-term fluctuations or variations in performance. There's an optimal amount of variability that you need to be able to adapt to ever-changing stressors and environments that these systems are placed in. Too much is indicative of a system that cannot stabilize, and too little variability is indicative of a system that go beyond just quantifying something like standard deviation and actually look at the predictability or self-similarity of these fluctuations. Now together, my hands, I like to gesture, I apologize. Together, they all fluctuate, and these fluctuations are not random. They are there and controlled physiologically in order to allow systems to adapt. So really understand cognitive function and brain health. And so understanding what those are and looking beyond just the basic interactions and how these are mediated by lifestyle factors and all kinds of other factors. So at risk for functional decline. So we have been collecting data preliminarily to understand really what's going on here. We've enrolled about 20 individuals between the ages of 45 and 85 years old, and they've gone through a variety of a battery of tests, but I'll talk just about the variables that we're most interested in. We've looked at walking speed reserve, which we define as the difference between baseline and maximal walking speed. Cardiorespiratory reserve, which is their difference between resting heart rate and then their maximal heart rate on a maximal cardiopulmonary exercise test. And then we also looked at the VO2, a measure of cardiopulmonary function between the start and end of that test. We've also looked at something that we're just generally calling cognitive reserve, and that's really the difference in bold activation between doing a lower difficulty cognitive test and a higher cognitive test. And we're using the Sternberg to change that difficulty there. Our preliminary results are very preliminary, and you can learn a lot more about the data that we have and what we're presenting here at this conference at posters 52 and 53 this afternoon, so please stop by. But preliminarily, we do see that walking speed reserve is related to the presence of plasma p-tau. Oh, I left that bit out. We also did measures of the ATN framework. We have amyloid beta, p-tau, and then total hippocampal volume. Feedback on this project. I'm newer to Texas, and improvement of early identification and early intervention. And to do that, predictive of future diagnostics, sophisticated math that measures your gait. Maybe you do this at a self-selected provider, can determine next steps if they're needed.
UT Health San Antonio.
So we've, let's see. I'm sorry. The screen. Okay, awesome. Sorry. So we've known for a while that atrophy and hypometabolism are distinct measures, both in time and location. So we know that atrophy occurs in the medial and lateral temporal areas, whereas hypometabolism occurs in the posterior cingulate cortex and temporal parietal areas. You can see this in this figure by Clifford Jack from 2010. So the FDG PET measures of hypometabolism are becoming abnormal quite a bit before. So this leads us to our goals of the project, which is to perform a large-scale meta-analytic characterization of atrophy and hypometabolism patterns in AD. So specifically, we want to determine which is a neuroimaging database aimed to help at meta-analytic analyses of imaging data. So from this, we gathered our data set of atrophy subjects, which was about 9,000 subjects. Our data set of hypometabolism subjects was about 10,000 subjects. With these, we found regional patterns of change, so where does atrophy occur and where does hypometabolism occur, using an algorithm called activation likelihood estimation, or ALE. And this just determines where the different studies converge above chance. From here, we looked at which behaviors are associated with regions that show atrophy, which behaviors are associated with regions that show hypometabolism. And again, we used data from BrainMap, this time from the healthy task activation data. So our next step was to look at the network level data. So you can see that atrophy here in red is mostly in the bilateral hippocampus, the bilateral temporal gyrus, and the insula, whereas you're seeing hypometabolism is largely overlapping with areas of amyloid deposition. So looking at what behaviors are associated with regions that we know are atrophied or show hypometabolism, the top 3 behaviors associated with atrophied regions are explicit memory, fear, and olfaction, whereas the top 3 behaviors associated with regions showing hypometabolism are regions. In this framework, we have a relatively simplified approach of just binary positive-negative measures for AT and N, whereas the recently updated 2024 framework, since it's kind of trending towards being a little shown, then that is pretty specific for Alzheimer's disease. Since atrophy and hypometabolism are shown in different patterns, we suspect that they may actually be due to different underlying pathologies. And since the goal of biomarker profiling is to represent an individual's pathology, we would recommend splitting neurodegeneration into N sub S for atrophy and N sub F for hypometabolism. So this brings us to our third finding, which is looking at these changes more at a network level. So both atrophy and hypometabolism in AD follow what's called network-based neurodegeneration. So the regions don't become abnormal randomly in spatial thresholding for each. So this brings us to our second recommendation that we would make, which is to use network structure as a biomarker. So specifically, you can use individual edge weights, you can use combinations of edge weights, as well as overall model fit, or the correlation of any of these with symptoms, and compare the differences, between healthy controls as well as people with MCI or AD. This type of analysis is currently being explored in various psychiatric disorders as well as MS, and so we think that this is something that AD could benefit from as well. So in terms of our main takeaways, so atrophy and hypometabolism are showing distinct patterns in AD, aligning with different behavioral changes. Because they're so distinct, we think they may be due to underlying pathologies. So we would advocate for the inclusion of these two as separate biomarker categories. And current work is looking at using the network models that we've developed and testing them in primary data for biomarker potential, so specifically for diagnosis and monitoring. Another potential implication of this is its use for neuromodulation target measures. So
with this, we conclude the first part of this session. We will have about a 30 minutes break. For the people that is having posters, please use this time to set up the posters. If you don't know the number where to put the posters, they are here. Just look your abstract and you will have the number. And I encourage you to talk with the speakers and visit the posters. And see you at 10.
We will now take a short break and meet back for the keynote address in Zlotnik at noon. We will now take a short break and reconvene at 10 a.m.
Actually, I don't know, but I think that
As we know, our population is rapidly aging. Dementia is an increasingly big problem. Everybody knows someone who's been affected by this condition.
We're not going to be able to cure Alzheimer's disease and related conditions in the very near future. And I think TARC has been instrumental in bringing a bunch of these new bright people into the field to kickstart their funding such that they become eligible then for funding at the federal level.
So they're really just getting their feet under them. They take the TARC money and put it to great use, and then continue to make an application for things at the federal level. Much, much more generous and larger dollar values are awarded.
It can be very difficult for somebody starting their research career to get enough funding to put their first set of...
I was fortunate to get one of the postdoctoral fellowship training grants and recruited a great early postdoc, and she has become an investigator, gotten her own grants.
It is not beyond the realm of possibility that he or she can turn that initial $300,000 or $400,000 into several million. And we are seeing today in TARC, in the real world, it is happening.
So if Texas has ways of using the seed money to cleverly coordinate these efforts, I think that it bodes well for the future.
Posters, and just to let you know, we will have a little bit shorter session in your agendas. If you want to mark that the last session will not be occurring because Dr. Ma excused herself due to the weather. Was bad in Houston. So we will start now with Dr. Maya Henry from UT Austin.
I'm going to be telling you about some of the work that we do here at UT Austin, looking at behavioral interventions to promote communication that we are working on. All of this work is funded by the National Institutes of Health, NIDCD, and NIA. So many of you are familiar with Alzheimer's in its most typical presentation where memory is the most significant challenge, but there is growing understanding that there are different dementia phenotypes, of course, and some of our patients present with prominent language impairment or speech impairment, and erosion of their ability to communicate is the most significant challenge that they face. And for a long time, these people were told that clinical nihilism, so we're investigating a variety of treatment approaches designed to rebuild spoken language, to work on other modalities and connect with the world. There are significant psychosocial ramifications, so we're thinking about counseling and other psychosocial interventions as well. And because these patients have no time to lose, we're interested in making treatment as efficient and potent as we can, so we're pairing our behavioral treatments with neuromodulatory interventions as well. We have a number of collaborators, so these are multi-sane as well, where they're investigating interventions for people who are bilingual. So just articulating words with speaking in sentences, this is just a snapshot of some. The most exciting thing, I think, is that, and we're hearing this from care partners as well, we collect baseline imaging. These interventions are also appropriate for people with other language prominent presentations of dementia, So we're seeing different patterns of brain regions that are predicting Alzheimer's disease. And these imaging findings are helping us to inform neurobiologically motivated models of brain stimulation to pair with our speech and language interventions. I should mention that we are delivering almost all of this treatment remotely delivered brain communication. So we're interweaving different treatment approaches, working on spoken language while we're working on other communication modalities. As I mentioned, interweaving things like cognitive with people who are bilingual and come from other cultures and different ethno-racial backgrounds. And Dr. Grasso is really spearheading that work. The fact that we can deliver interventions to people in their home allows us to see them wherever they are. So to circumvent whatever mobility limitations that they have, and also to reach a more diverse sample. Some bigger picture perspectives. As I said, these patients with a language-led dementia have been sort of underrepresented in the treatment literature, and we've lacked evidence to support intervening with them, but we have a growing evidence base now that not only can they respond to speech and language treatment, they can recover their ability to and don't have control. But the big picture here is really that this kind of treatment really should be the standard of care for these patients and not the exception. We're still seeing the little...
The next speaker is Mr. Jose Rios from UTRGB.
Excuse me. Okay, good morning, everyone. My name is Jose Rios. I'm a research associate for Dr. Khalid Benamar over at UT Health Rio Grande Valley. And today we'll be discussing Alzheimer's disease and HIV and comorbidity. So without further ado, let's get started. Causes of neurodegenerative dementia being characterized by a progressive impairment of cognitive functions. So some pathophysiological characteristics of Alzheimer's disease. 14 years following PET imaging then demonstrated some marked cortical amyloid beta plaque deposits. And the second case, a 70-year-old woman with HIV encephalitis presented with memory complaints. Following PET imaging as well detected the abnormal phosphorylation of the tau protein. So here, aging is the primary risk factor for Alzheimer's disease, and 53% of American adults living with HIV are over the age of 50. So as the population ages, the number of individuals who experience Alzheimer's and HIV will also increase. So therefore, the comorbidity of these two clinically unmet is unavoidable. So for experimental reduces aspects of HIV and the mouse model. So this is a recently established Alzheimer's disease mouse model in which 3 amino acids within the amyloid beta region are knocked into the mouse APP sequence to express human amyloid beta. And the second mouse model that we use is the tau. We use both of these models to see if these models potentially show if HIV favorably interacts with either amyloidogenesis or tauopathy to accelerate the decline trajectory of Alzheimer's disease itself. So the test that was used on the right-hand side, as you can see, it is called the Novel Object Recognition Test. So this test is performed in an open field box, and it is a test that relies on the rodent's natural tendency for exploring novelty. So before any of the testing began, starting on the first image, the mice each that were tested were habituated in the arena for 5 minutes without any objects for the first day. So we used this day to let them get kind of comfortable with the environment just for accurate data result. So after this 24 hours, after the habituation period, each mouse was placed into the same arena and now placed with 2 identical objects and is used as the familiar. This is a 10-minute test for each animal being tested. So the time spent exploring each object was then recorded using a video tracking system, and the experiments were of course done blindly to generate the data that we will discuss in the next couple of slides. Okay, so now on to results, starting with the tau mouse model, and as you can see, the eco-HIV promotes the onset of cognitive decline in the tau mice at 5 weeks post-eco-HIV injections. Starting from the left-hand side, the data collected is shown by the total investigation time, which is calculated from the time spent on the novel object divided by the time spent on both the novel and the familiar object, all multiplied by 100. So, and as you can see in the first 2 graphs for the eco-HIV, it has still yet to promote the cognitive decline until on the very right-hand side, until comparing it to the post 5 weeks, when we finally start to see the eco-HIV promotes, as in the previous slide, the onset of cognitive decline, but this time at 11 weeks post-eco-HIV injections. So for this, starting with graphs A through graph F, where it shows the eco-HIV finally kicking in to promote the cognitive decline. So unlike the last slide, all of this data is collected, showing the total investigation time period. And as for the amyloid beta mouse model at the right, to be the first evidence for an in vivo function. Thank you.
Next, we will have Dr. Hannah Hausman from UT Health San Antonio.
There we go. Good morning, everyone. I'm Hannah Hausman. I'm a neuropsychology fellow at UT Health San Antonio, and I'll be presenting on evaluating a two-step cognitive screening process. Let's see if it'll go forward here. Yes, I'm trying to. Oh, there we go. So dementia is a growing public health concern, as we all know, with the prevalence of Alzheimer's disease expected to nearly double by the year 2060 in the United States. Early detection of cognitive impairment is important for improving patient outcomes, reducing. Primary care settings are well positioned to detect at-risk individuals for cognitive impairment because they may be the first to hear of cognitive concerns. Setting, as only about 16% of older adults report receiving a cognitive assessment as a part of their health care. There are barriers to engaging in early detection of cognitive impairment. From a provider standpoint, they can report concerns regarding hesitancy in choosing, administering, or interpreting different cognitive tools, which there are many, as well as knowing the appropriate next steps following a positive cognitive screening results with regards to referrals, workup, or their own role in dementia care. There are also its benefits. And then from a provider standpoint, again, they may not be aware of who is an appropriate candidate to receive a cognitive screener at these visits. Given our growing older adult population, it's not feasible to first identify a screener could also lead to that monitoring and reevaluation, while a positive cognitive screening result would trigger the need for further assessment. So first we looked at the effectiveness of a risk assessment tool called the Brief Dementia, or if they need assistance in their ADLs. Then we aim to examine if combining the risk screening measure with performance on common cognitive screeners would improve the classification accuracy of cognitive status as either normal or impaired. In this study, we use a two-step approach that's administered on an iPad and combines are appropriate. But for the results that I'm presenting on today, we recruited 150 in the QMCI. First, examining the effectiveness of the BDSI alone in identifying risk impairment amongst our cognitively normal and cognitively impaired individuals. We see that the BDSI classified 70% of those with cognitive impairment, saying that they were at low risk for impairment. Next, we then combined the risk status from BDSI and their performance on the common cognitive screeners, and overall adding this screening information, a similar pattern of having low false positives but high false negatives. Comparing the two combined model picture, a success of this work is starting to create a foundation for looking at a two-step approach to improve that early detection of cognitive impairment within a primary care setting. Incorporating a risk assessment, there is a need to improve the sensitivity of those risk assessment measures administered at that first step to make sure we're fully capturing those with cognitive impairment. This could include investigating a combination of risk assessment approaches, refining or adding test items, looking at various cut points, or even analyzing risk as a continuous outcome. Altogether, if we can work to refine this two-step approach, we could transform dementia care by addressing gaps in the early detection of impairment, allowing patients and their families to take action earlier, engage in timely intervention, and hopefully improve the level of support, enhancing their quality of life. Thank you all for listening. I'd like to thank our collaborators, Drs. Roop, Hilsebeck, and Davis, and enjoy the rest of your day. Thank you.
Now we will hear from Dr. Manuel Silva Perez from Baylor College of Medicine.
Good morning, everyone. I'm excited to be here. My name is Manuel Silva Perez, and I'm a postdoc associate at Ginny Chin's lab, Baylor College of Medicine. We study AD through mouse models of AD neuropathology, and my work in particular focuses on the link between AD, sleep, epilepsy, and norepinephrine. Alzheimer's disease, as you all know, has many pathological manifestations such as memory problems, neurodegeneration, sleep disturbances, epileptic activity, attention and arousal problems, anxiety, or inflammation. Norepinephrine is an important neuromodulator that is implicated in many of these same domains. Particularly, it has neuroprotective, anti-epileptic, and anti-inflammatory properties. The overlap between AD hallmarks and norepinephrine's effects led us to wonder whether norepinephrine dysregulation may underlie or be a cause of some of AD's symptoms. And the main source of norepinephrine in the brain is the locus coeruleus, and indeed, there's more common in AD patients than in the general population. And interestingly, norepinephrine in this context would have therapeutic effects for this desipramine and reboxetine. And what do these two compounds do? So as norepinephrine is released to the extracellular space, it builds up and exerts its effects. And then it is actively taken back into norepinephrine axons due to transporters. What these compounds do is blocking these transporters, and it's important because in AD, most of epileptic activity happens saline administration. Mice had plenty of epileptic spikes, especially during sleep, shown here in blue and purple, but they had almost nothing during wakefulness, shown in orange. Epileptic spikes are these large downward deflections of the signal and are also indicated with vertical lines above. But upon injecting IP reboxetine or desipramine, there was a strong acute reduction in spikes, which gradually came back several hours later as the drugs were metabolized. Here is a quantification of this phenomenon over the 12 hours after injection. So for the first block of 4 hours, we saw a strong reduction in spikes that gradually disappeared as the drugs were metabolized. This is a quantification for non-REM sleep in particular, which is, again, where most of epileptic activity happens in AD. So we found that, indeed, norepinephrine has anti-epileptic effects in the context of AD-related epilepsy. But we wanted to confirm that it was indeed due to enhanced norepinephrine and not to non-specific side effects. And we used, we found confirmation using a chemogenetic approach. For that, we expressed excitatory dreads in the locus coeruleus of APP mice, implanted them for to be able to specifically activate norepinephrine neurons. We implanted them for EEG recordings, and then administered either of two very selective agonists to be able to stimulate the locus into releasing more norepinephrine. As you saw in the previous slides, in controlled conditions, upon injection of saline, the mice had plenty of spikes, specifically during sleep. However, on the other hand, upon injecting them with the two specific agonists, we saw a reduction in spikes that gradually went back to baseline. And here again is a quantification of the phenomenon. We saw a strong reduction for 4 to 8 hours, which disappeared as the drugs were metabolized. So these results were really exciting. And we wonder whether chronically enhancing norepinephrine could have disease-modifying effects. To check that, we implanted APP mice with Alzheimer osmotic mini pumps designed to deliver either reboxetine or saline for control over plaque load developed in hippocampus and cortex. On the other hand, upon treating the mice with reboxetine for 28 days, we saw reduced plaque load in both areas. This phenomenon is quantified here. We saw a strong reduction at 65 and 71%. That red data points indicate the mice shown in the indeed has anti-epileptic effects in the context of AD, and it could have exciting therapeutic potential. AD. And lastly, I would like to thank our lab members for their support and camaraderie, and also thank our funders and TARC for travel funds and also for the opportunity to present here. Thank you very much.
Good morning, Alzheimer's Association. We'll start the talk with some nomenclature. Lewy body disease is an umbrella term that they're caused by an abnormal buildup of as disease manifestations in the Lewy body disorders. Although the visual hallucinations are very common in Lewy body disorders, affecting 60% of people with Parkinson's and 80% of those who have visual hallucinations, such as seeing people or animals in the room that are not actually there. And despite how prevalent this problem is in these disorders, the treatment options are really quite limited. because most of the medications we have available to us to treat hallucinations, i.e. antipsychotics, can cause worsening of the motor symptoms of Parkinson's, like tremor, stiffness, slowness, falls. So we're limited to 3 main options: quetiapine, pimavanserin, and clozapine, that have less dopamine blocking effect. And the 2 first-line ones are considered quetiapine and pimavanserin, because clozapine can come with some serious side effects and is difficult to prescribe. We have larger and more positive trials, but it's worth noting that this population is very hard to recruit